Okay, our next speaker is Dr. Arun Shukla, who's an associate professor at IIT Kanpur. He's recipient of EMBO Young Investigator Award and the Intermediate Fellowship from India Alliance. Arun's interest lies in studying structural and function of GPCRs.
It's an absolute pleasure to be here. As we are celebrating a decade of India Alliance, I thought I'll give you a brief overview of the research program in my lab that we have established in the last nearly half a decade. And I'll not bog you down with details of experimental data, but I'll be available in lunch if anybody wants to talk about specifics. So we are primarily interested in a family of receptors called G-protein coupled receptors, GPCRs, and they constitute the largest family of receptors in our body, about 1,000 different members. They're also referred to as 7 transmembrane receptors because they span the membrane 7 times. And about 200 or 250 of them are functionally known, while the remaining receptors still remain orphan. And these receptors are involved in every physiological process that you can think of, starting from brain function, behavior, cardiovascular regulation, pain sensation, immune response, and so on. And thus, it's not surprising that about 40% of the currently prescribed drugs that target GPCRs. For example, angiotensin receptor blockers are prescribed for hypertension, histamine receptor antagonists for allergies, long-acting beta agonists for asthma, and so on. Therefore, it's very important to understand how these receptors work. That is, how they recognize ligands, how they're activated, how they signal, and how they're regulated, not only to understand the basic aspects of cell signaling, but also in long term to utilize that information to design better therapeutics. So I'll take you through the current paradigm of how these receptors work based on several decades of research. So binding of an agonist, which is an activating ligand, leads to a conformational change in the receptor and its activation, followed by coupling of heterotrimeric G proteins, and hence the name G protein coupled receptors. This, in turn, results in generation of second messengers, such as cyclic AMP, inositol phosphate, calcium, leading to what we typically refer to as G protein dependent signaling. But obviously, cells have to control the signaling in a very precise manner. And for that, they have designed a mechanism called desensitization. Essentially, what happens is that activated receptors are phosphorylated primarily in their carboxyl terminus by a family of kinases known as GPCR kinases or GRKs, and that leads to interaction of a multifunctional protein called beta-arrestin. You'll be hearing more about this in today's lecture. And it is believed that interaction of beta-arrestins essentially comes in the way of G-protein coupling. That is, they sterically hinder further G-protein coupling, leading to termination of G-protein signaling response. Now, this has been the classical paradigm. However, in the last 10, 15 years, it has been discovered that beta-arrestins can do many additional things. For example, they can interact with a number of components in endocytotic machinery, such as clathrin and adaptin, to mediate receptor endocytosis, which is basically the process of receptor trafficking from the cell surface to inside the cell. They can also scaffold E3 ubiquitin ligases to bring them in close proximity of the receptor, thereby mediating ubiquitination and degradation of the receptors. But most importantly, they can also scaffold a number of kinases and phosphatases to yield what we refer to as G-protein independent, beta-arrestin dependent signaling. So essentially what it means that, unlike what is there in the textbook, the signaling downstream of GPCRs is bifurcated. It can happen through G proteins or it can happen through beta-arrestins. And an interesting aspect of this is the cellular and physiological outcomes downstream of G protein signaling or beta-arrestin signaling, they're very distinct from each other. And that has huge applications because in the past few years, it has been possible to pharmacologically separate these two signaling pathways. And I'll tell you what I mean by that. So when an antagonist, which is a ligand which inactivates the receptor or puts it in inactive conformation, none of these signaling pathways are triggered. When a full agonist or unbiased ligand or endogenous ligand for a receptor, when it binds to a GPCR, you get both arms of signaling G proteins and beta-arrestins. But it is possible to design ligands which will bind to the receptor and selectively trigger either beta-arrestin pathway or G-protein pathway. And these ligands are referred to as beta-arrestin biased or G-protein biased ligands. And it turns out that many drugs that are prescribed that work on GPCRs, their side effects and their desired effects are going through either the G protein or the beta-arrestin pathways, meaning the desired effect and side effects are separable from each other. And this has sort of led to the concept of biased agonism that has implications for novel therapeutics, and I'll just give you one quick example. So morphine, which is taken to manage post-surgical pain, is basically an unbiased ligand for mu-opioid receptor, or GPCR. It's very effective analgesic, but it also leads to undesired effects of constipation, respiratory problems. It turns out that the pain relief is going through G protein pathway, while the undesired effects are going through beta-arrestin pathway. So now if you think about it, if you can modify morphine or design new analogs or identify new ligands from scratch, which are G protein biased ligands, if they bind to the receptor, they will give you pain relief that you want, but not the side effect. And hence, what you have is a more effective therapeutic, so to say. And this is one example. There are multiple examples across many different receptor systems. In some cases, it's the G protein signaling that gives you desired effect. In some cases, it's beta-arrestin signaling that gives you the beneficial effects. And now this is changing the landscape of drug discovery in major pharmaceutical companies that are focused on GPCRs, and many of the biased ligands are in different phases of clinical trials, including one that is in the final stages of approval by FDA. So receptor G protein interaction has been relatively well understood, but receptor beta-arrestin interaction and the subsequent regulation of the receptors is lagging behind, and that is where our focus has been in the last 4.5, 5 years or so. So this interaction of receptors with beta-arrestins is a biphasic process. So what happens in the first step, beta-arrestin, which is shown here in blue, interacts with the carboxyl tail or carboxyl terminus of the receptor, shown here in yellow, and forms what is referred to as partially engaged complex, or also referred to as tail-engaged complex. And then in the second step, beta-arrestins engage further with the transmembrane bundle of the receptor, leading to the formation of fully engaged complexes. And in the field, it has been generally sort of believed that the fully engaged complex is the one that mediates all the functions of beta-arrestins, and the partially engaged complex is simply a passive intermediate. But when we started, we didn't sort of buy this notion, and we used a combination of pharmacological, biochemical, structural approaches to assemble these two types of complexes in vitro and in cellulo, and ask the question whether the partially engaged complex might also have some functional relevance. And turns out that in fact there is a functional separation in these two conformations of receptor beta-arrestin complexes, where the partially engaged complex is competent to mediate endocytosis and signaling, while the fully engaged complex is driving the desensitization. And when you think about it, it makes sense, because for desensitization to happen, beta-arrestin has to snuggle against the receptor so that it can occlude the surface on the receptor, which was otherwise being occupied by G proteins. And partially engaged complexes can drive endocytosis and signaling, because beta-arrestin, even in this conformation, can interact with clathrin, can interact with ERK-MAP kinases, and so on. Now, since we published it, 3 other groups across the world, they have, using different receptor systems, different approaches, they have converged to the same notion, suggesting that this is a new paradigm in these complexes, how these complexes work. Interestingly, what we have also found is that if we take a beta-arrestin bias ligand, which upon interaction with the receptor mediates only beta-arrestin signaling but not G protein signaling, it exclusively forms only the partially engaged complex, and there is no formation of fully engaged complex. And it makes sense, because in this case, when a bias ligand binds to the receptor, there's no requirement for desensitization, because there's no G protein coupling in the first place. Whatever is required of the ligand, that is endocytosis and signaling aspect, can be mediated by partially engaged complex. So it provides a previously lacking mechanistic insight into biased signaling and how biased ligands mediate their effects. Now, what I have told you so far, basically this paradigm has been developed using synthetic ligands or receptor mutants. So a question is whether, if we can sort of design and understand it in vitro, is it possible that nature has already designed it in our system? We have just not identified them. Meaning, is it possible that bias ligands, bias receptor systems might be available or might be present in our bodies? And when we look to the literature, it turns out that there are many 7-transmembrane receptors which are classified or which are still present in the GPCR superfamily, but because they have 7-transmembrane architecture, a typical signature of GPCRs, but they do not couple to heterotrimeric G proteins, and hence they're basically classified and somewhat ignored as non-signaling or non-functional receptors. But if you knock them out, you get very specific phenotype, suggesting they must have some functional role. Intuitively also, it doesn't make sense that nature would design something that's sitting in the membrane, taking space and doing nothing. So we basically, and at the time when they were classified as non-signaling receptors, the beta-arrestin signaling was not appreciated that much, and hence people thought the only way these receptors can signal is through G proteins, and hence they were classified as non-signaling receptors. So we have basically connected these two dots and asked the question whether these so-called non-signaling or non-functional receptors, might they be signaling through alternative pathways, perhaps beta-arrestin pathway. And in fact, what we have found is, as present in the literature, these receptors do not couple to heterotrimeric G proteins. They are basically flat in G protein coupling assays. But they do very robustly recruit beta-arrestins. Not only they physically interact with beta-arrestins, but they're also getting internalized in beta-arrestin dependent fashion, meaning they are in fact beta-arrestin coupled receptors. What about their signaling? And to identify, to discover whether these receptors might signal, and if they do, what signaling pathways might be involved, we have used antibody array-based approaches or global phosphoproteomics analysis to identify different proteins which undergo phosphorylation, dephosphorylation, when these receptors are activated. We have identified a number of proteins. Some of them fall in the previously known category of beta-arrestin signaling pathways, and we have identified some unique pathways as unique proteins as well. And this phosphoproteomics was done with vProteomics, a venture in Delhi that is started by a former India Alliance fellow, Gagandeep. And in the past, we have worked with top-notch ventures in the world, and the data quality, the services that they provide is on par with international ventures. So we are very happy sort of in working with them. And once we have identified these signaling proteins downstream of these beta-arrestin coupled receptors, we have utilized basic bioinformatic analysis and then identified new signaling pathways that some of these receptors can signal through. And we have experimentally validated some of them using inhibitors or knockdown approaches. Interestingly, when we reconstitute beta-arrestin complexes of these receptors using stabilizing antibody fragments, what we find is that these receptors exclusively form this partially engaged complexes with beta-arrestins. So it's coming back to the biased ligand scenario, where biased ligands induce only partially engaged complex. Similarly, these receptors bound to their native or endogenous ligand, they also induce formation of only partially engaged complexes. And this makes sense, again, because similar to biased ligands, there's no G protein coupling for these receptors, and hence there is no need for desensitization, and therefore there is no requirement for the fully engaged complex. Now, going forward, we are using cryo-EM and X-ray crystallography to get high-resolution structure of these receptors to see, despite having 7-transmembrane architecture, how they can display such a unique functional divergence compared to prototypical GPCRs. Now, I've been using the term beta-arrestins, plural, because there are two isoforms of beta-arrestins, beta-arrestin 1 and 2. And if we superimpose their structures, their three-dimensional structures, if these were not labeled, even I cannot tell which one is beta-arrestin 1 and which is beta-arrestin 2, because they're structurally very similar. They have about 78% sequence identity. Interestingly, it turns out that they have, despite having structural and primary sequence similarities, they have a very significant degree of functional divergence. And what I mean by that is, for a given receptor, it's possible that beta-arrestin 2 is predominantly mediating endocytosis, while beta-arrestin 1 is mediating signaling, or vice versa. So that is one receptor, two isoforms of beta-arrestins doing different things. There are also cases where the same isoform of beta-arrestins can mediate very distinct signaling pathways for different receptors. So how does it work? And this has been a huge mystery in the field. And we have basically sort of utilized, once again, a combinatorial approach of structural, biophysical, biochemical, sort of techniques, including spectroscopy, negative staining EM, to identify that the two beta-arrestin isoforms, that is beta-arrestin 1 and 2, when they bind to the same receptor, they can adopt very distinct conformations. And in fact, these distinct conformations, in turn, drive their functional divergence. So we can make chimeric beta-arrestin constructs, and we can encode new functions in them. What I mean by that is we can make beta-arrestin 1 look more similar functionally to beta-arrestin 2 and vice versa. So there's distinct conformations of beta-arrestins that they can adopt when in complex with a GPCR is driving their functional divergence. So this has come back with decent reviews. We are hoping to send it back in a few weeks. What about same isoform doing different things for different receptors? And to probe that, we have designed antibody fragment-based sensors that can selectively recognize receptor beta-arrestin complexes in vitro and in cellular context. And I'm just giving one example here. So in green, you basically have an antibody fragment that is expressed as an intrabody. An intrabody is basically when you express an antibody as cytoplasmic protein. And the intrabody is tagged with YFP. Beta-arrestin 1 is tagged with red and cherry. And this intrabody has specific recognition for receptor-bound beta-arrestin 1. So what we see here is that under basal condition, they're both cytoplasmically distributed, as expected. When we hit the cells with agonist, beta-arrestin from cytoplasm goes to the membrane, where the receptor is sitting, so that it can interact with the receptor. And intrabody very faithfully follows the receptor to the membrane. Upon longer agonist exposure, the receptor beta-arrestin complexes get internalized in these endosomal vesicles. And once again, the intrabody sensor follows the complexes in these endosomal vesicles, suggesting it's a good sensor of receptor beta-arrestin interaction, their trafficking, and the complexes that are formed in the cells. Now we have used this intrabody sensor and looked at about a dozen different GPCRs. And turns out about half a dozen receptors behave the same way. That is, they form a type of complex or a conformation with beta-arrestin that can easily be recognized and followed by the intrabody sensor. But then the other half a dozen or so receptors, including, for example, bradykinin receptors, they show a very distinct pattern. So in this case, bradykinin receptor is a prototypical GPCR, like vasopressin receptor, which was used here. Again, upon agonist stimulation, beta-arrestins go to the cell surface, and then they get internalized in these endosomal vesicles with the receptor. But unlike vasopressin receptor here, the intrabody is sitting pretty in the cytoplasm even after agonist stimulation, suggesting that the complex of beta-arrestin 1 with bradykinin receptor must be conformationally very distinct from the complex of vasopressin receptor beta-arrestin 1 complex. So this sort of provides a mechanistic insight into how same isoform of beta-arrestins can mediate different functions for different receptors because they can form different conformations with these receptors. Now, my talk title said structure, function, and modulation, so I'll talk about modulation in 2 minutes. So as I said, the driving force for the functional multiplicity of beta-arrestins has been their ability to interact with a number of cellular partners such as clathrin, ERK MAP kinase, JNK, CSORC, and so on. So we have, and if you look at the interface of these interactions, they are very distinct on beta-arrestin's surface. And therefore, we wondered whether it would be possible to selectively interfere some of these interactions and thereby selectively take out one function of beta-arrestin while not affecting the others. If you think about siRNA knockdown, knockout approaches, CRISPR-Cas, they basically lower the level of protein, such as beta-arrestin, lose all the functions across the board. We wanted to selectively take out one function while not affecting the others. So again, we have used phage display-based technology platform to generate antibody fragments against beta-arrestin 1, beta-arrestin 2, and so on. And I'll just give you one example. We have identified a particular antibody fragment that very selectively inhibits the interaction of beta-arrestin with clathrin. It does not significantly affect the interaction of beta-arrestin with ERK MAP kinase. When we express it as an intrabody in cells, it can very efficiently inhibit the endocytosis of vasopressin receptor, which is a prototypical GPCR, shown here by whole cell ELISA, also by confocal microscopy, where in case of control antibody fragment, the receptor is internalized in these endosomal vesicles. In case of cells expressing this inhibitor of beta-arrestin-clathrin interaction, receptor beta-arrestin complexes remain tethered to the membrane even after prolonged exposure. So this has allowed us to separate the endocytosis and signaling from each other, which sort of changes the previous notion in the field that you must have endocytosis to get ERK-MAP kinase activation in beta-arrestin dependent fashion. So earlier the idea was that you must have these signalosomes on the endosomal vesicles to get ERK-MAP kinase activation. But using this antibody fragment as a tool, we have sort of defined that separately. And interestingly, as I said earlier, it has also been possible to use this antibody fragment to selectively take out endocytosis function without affecting ERK-MAP kinase activation. We have now antibody fragments which can do the opposite, that they can inhibit ERK-MAP kinase signaling without affecting the endocytosis. And more interestingly, we have identified antibody fragments that can allosterically stabilize these complexes in the cells, and thereby they can enhance, for example, the kinetics or the onset of signaling or endocytosis, and we can play with compartmentalization of signaling and so on. Hopefully, I'll be able to tell you more about that next time. So due to time constraint, I'll not talk about the summary, but in 2 minutes, I'll tell you about the impact of India Alliance as I see it. Since I moved to India, on a couple of occasions, I've been asked by different outlets to share my experience and why I decided to move back to India. And I've said what I honestly feel. If it was not for India Alliance Fellowship Program, perhaps I would have not returned back to India in the first place. And even if somehow I did, perhaps we could not have done whatever little we have done in the last 1.5 years. But in my honest opinion, my personal opinion, I think the impact of India Alliance is not about the success of individual fellows or a few fellows or some of us publishing glossy journals or awards and so on. But rather, the impact is that it has inculcated a belief, a faith, a confidence in us that despite all odds, we can do cutting-edge, globally competitive science in India. And I think that is the true impact. The other thing which I feel the Alliance has done is it has broadened the footprint of quality science across the country. So it's not the case that you get quality science only in localized pockets, but now it's spreading all over the country. What really gives me immense pleasure when I go to a Gordon meeting or Keystone meeting, when a very productive postdoc comes to me and says, I was thinking about setting up a shop in States or Europe, but now after seeing what India Alliance fellows can do in India, I have decided to apply in India and look for a faculty position there, and that is what makes me ecstatic. Obviously, the last decade has been phenomenal of India Alliance, but I can say with full conviction that the next 10 years are going to be even more productive, more exciting, and take the Indian science to a whole new level. As they say in Hindi, kahani abhi baki hai. Thank you.
Thank you, Arun, for a very wonderful presentation and very kind words. So the hall is now open for questions. We can take only a couple of questions in the interest of time, Professor Shankar.
Thank you for this really interesting talk. So, have you looked at the classification of this non-canonical GPCR beta-arrestin signaling across different cell types to see if they are A, cell type specific, or if they are a given function, phenotypic function. I'm not talking about phenotypic function in terms of ERK signaling, I'm talking phenotypic function in terms of the endpoint consequence in cells. Have you looked at the classification of these under relevance and cellular function and normal and pathological function?
Sure, absolutely. So that's a wonderful thing, and we are sort of going in that direction a little bit. For example, the signaling pathway and the cellular outcome that we have identified for one of the chemokine receptor D6 is, it seems to be in a PKD, AKD, coffulin pathway in migration. And these receptors are expressed in those immune sort of cells where migration is one of the key functional outcomes. So that does seem to make sense. But we have not sort of looked at the entire family of receptors yet, because the idea, at least in the beginning, was to establish that these are indeed beta-arrestin signaling receptors and then see how wider, how many other receptors will follow the same pattern. So going forward, we'll definitely be looking at that.
Okay, one last question there.
I have a short question. See, how stable are these intermediates and physiologically? And your antibody fragments are actually interfering with its stability when you track the protein in or out?
Right. So that's a very good question. In fact, for structural studies, we have to stabilize these complexes, even the fully engaged ones, with antibody fragments. Otherwise, we cannot really capture them by crystallography or by cryo-EM. But that is the beauty of the biased ligand and the biased receptors, where all that we are getting in cellular context as well, without any antibody fragment stabilization, is only the partially engaged complexes, meaning that these are reasonably stable. If you compare them side by side with fully engaged in terms of kinetics and stability, that we have not done, but they seem to be reasonably stable where they can mediate the functions downstream.
Okay, thank you, Arun.